Welcome BIS2B students. Today we'll be studying genetics. We'll start with a brief introduction. For some of you, this may be more of a review, but it will let us all start on the same page. You and I are comprised of many cells. The cells that we are comprised of are known as somatic cells, or body cells. Somatic cells all contain two sets of chromosomes. Each set of chromosomes in your body is inherited from one parent. Because there are two sets of chromosomes, these cells are known as diploid cells. The cells that we reproduce with are haploid. They only contain a single copy of our DNA. These reproductive cells are known as gametes, or sex cells. Examples of gametes are ova and sperm. To help you better understand chromosomes, we have model versions made with beads and the guidance of Dr. Pat Randolph.
For the first exercise in the lab, the goal is to reinforce concepts of genotype, phenotype, alleles, and gamete formation. And to do that, we would have given each student a set of beads here, where there's two pairs of homologous chromosomes. And you can identify the different pairs by the big white beads on one end and the square beads on the other. We've recreated this online, but I just want to quickly go over this to provide you some context. So here we're interested in two genes. On this pair of chromosomes here, there's a gene for head shape, and that's indicated by these two pink beads. But another possibility, if I go to this set of beads here, is there's a red version as well of that gene. Then we have a gene for foot temperature. That's indicated by these beads here. Then if I go to this set over here, this is also a gene for foot temperature. The different colors are representing alleles, or different versions of the gene. So in some cases here, they have the same copy of that gene, so it's homozygous. On this case here, there's a red and a pink, so there's two copies. This individual is heterozygous. So the idea here is, here's my genotype, represented by these beads. Translate that to a phenotype. So what does it mean if I have two pink beads and a green and a yellow bead here for foot temperature? Or what does it mean if I'm heterozygous for the head shape, and then I have a light blue and a dark blue bead for foot temperature? So the lab exercise online will walk you through that process. The second part of this is gamete formation. So remember, gametes are haploid cells. The sperm and eggs that we're talking about animals. And so to form gametes, we're going from a diploid cell, and this represents our diploid organism here, to haploid cells. To do that, recall during meiosis, the chromosomes will line up at the midline, and then they'll sort. So these guys would sort in this direction, these guys would sort in this direction. These would form one egg cell, for example, and these would form another egg cell, for example. So haploid cells, only one copy of each chromosome, resulting in haploid cells. I could do the same thing with this set here. So they line up at the midline, then they sort into different cells. That's one egg, that could be another egg, so creating the haploid cells. It is possible, if I go back to this set of beads here, as I sort the chromosomes, these two beads could have traded positions here, where in this case the red is going to be paired with the dark blue, or the pink with the light blue, and these could have sorted into a gamete, and these could have also sorted into a different gamete. And that's Mendel's laws of independent assortment. The idea being, if I go back to the starting point here, the light blue could end up with the pink, the dark blue with the red, or vice versa, the dark blue with the pink, the light blue with the red. And that's independent assortment. So then what you would do, and you'll get a chance to do this online, is you create your gametes, and then bring gametes together, two different individuals. So if I bring these two gametes together, I've recreated that diploid cell. So there's the offspring. What's the genotype of this offspring? Can you think about that? And that's Mendelian genetics. And again, you're going to have practice with that online.
For the first lab exercise, we have digitized the beads for you, but the context that Dr. Pat Randolph provided is still the same. Remember, if you have trouble with any of these concepts, please contact your TA. Below, you may begin exercise 1. Thank you for watching.